was about to start off this video saying I was going to show you one of my favorite services at BV-BRC. And it's true, this is one of my favorite services, but a lot of them are my favorite services. But I think you'll really like this series of videos because the variation service is pretty powerful, and the example, the data set is totally cool that we're going to look at too. So let's get started. So the type of data that you can submit for analysis in this service, it accepts long and short reads or sequence read archive run numbers. Those start with SRR or ERR if it's coming from the European nucleotide database. You can do multiple reads simultaneously. This thing runs really fast. You can really build it up. But let me warn you, if you put too many things on it, at the downside, when the results return, it generates a lot of results, and sometimes it's difficult to sift through. So I generally prefer like 3 or 4 at a time, but you know what? You be you, and you can do as many as you want. Now, the target genomes, these have to be annotated in BV-BRC. And the reason for that is we have the genome browser, the circular viewer. We have links to all the proteins, links to the conserved domain database, and it gives detailed analysis of the SNPs based on the annotations that are in BV-BRC. Now, those can be your private genomes, they can be public genomes, they can be the reference and representative genomes. Any of those, you can use those. There are, at this time, 5 types of aligners that you can use: BWA-MEM, BWA-MEM strict, bowtie 2, last. Last is the only one of these that's for long reads. Everybody else is short reads. And mini-map 2. For our particular example, we're going to use BWA-M strict because all of these genomes are really close together. So it's exposing an E. coli to antibiotic, and it's adjusting. So they're all related. Then there are the SNP colors. There are 2 of them that we have at BV-BRC, Freebase and BCF tools. Now, sometimes these will change. We may bring in different ones. We just recently added BCF tools. So, you know, sometimes things change right now. These are the 2 we have, and I'm going to use Freebase. You can store your data, or you have to store it in your private workspace. So you have to have your output folder and your output name, and then you can submit the job. And this is what the landing page looks like for the service app. Now, there are a lot of results that it generates. Let's say you do 5 different read files against the genome. You'll get repeats of 5 of these, so it will scroll on and on and on, but it gives you a lot of details that we're going to go over in other results. But some things you can do with that. It's going to give you a detailed SNP report telling you what gene or intergenic region, where that SNP occurs, and how it affects the gene. Does it totally knock it out? Is it just a change in the sequence? Is it a synonymous change that doesn't even affect the protein sequence? We show you how you can see these things on the genome browser, see exactly where they are, and then you can see the reads and how conserved those reads are across the read file, so you can see the read depth. We also allow you to visualize the SNPs on the circular viewer. So that's pretty sweet too. And in our public genomes, not your private genome, but in the public genomes, we use AlphaFolds. So you can map where these SNPs are occurring on a structure. We also have links to other resources like the conserved domain database at any of the gene pages, so you can see if there are any domains that are of interest. And with anything that we do, we want you to be able to find help. So there are ways to find information. There are user guides that are the information icons. There is a tutorial, and there will also be videos when I complete this set of videos. So clicking on the user guides will open a drop-down box that will give you different information about things. Clicking on that tutorial will show you all the information, including the citations for the different services. And I encourage you to go through that and scroll through these things, because some of the tables are pretty complicated, and it gives you detailed information on those. The data we're exploring is this awesome study by Bain et al. In 2016. This is a publication out of Science. And what they did was they created a giant agar plate. They put in different levels of antibiotic. In this case, it was trimethoprim. And they watched the organism, the E. coli, MG1665 is what they used. And they recorded it, and you can see it evolving in real time over 11 days. There's a great video on YouTube for this. If you just look up mega plate and antibiotic, it's one of my favorite videos. You should check it out. So this is what it looks like. This is the exposures that they had from absolutely no antibiotic at these ends, all the way up to lethal doses, all of these, and you can watch it evolving. And so every place where there's a little circle here, they plugged the agar and got the bacteria from those, and then they sequenced it, and all of those sequences are available in the Sequence Read Archive, and I have looked at all of them. Just a quick look on the action of antibiotics on bacteria. They can inhibit nucleotide synthesis, protein synthesis, or cell wall synthesis. Today, we're going to be looking at trimethoprim, which actually inhibits folate synthesis. And what happens is they do two different antibiotics, but trimethoprim acts here, where dihydrofolic acid is going to tetrahydrofolic acid. And that step requires NADPH going to NADP. I always like to look at the structures. I like them so much that I force you to look at them as well. So the trimethoprim and the dihydrofolate reductase share this particular structure here. And so that's where the magic happens, so to speak, if you're trying to knock out a bacterium. From the bacteria's point of view, that's where the devastation happens, or you conquer all. I looked, as I told you earlier, at all the mutations in this particular gene and mapped them and looked at all of them. And so that's where we're going to use a few of these samples. I have them all. I just really enjoyed this analysis and looking at them in detail. And I want to dig deeply into that one gene. There are other SNPs across the genome, but this is the one we're going to dig into. And one of the reasons I wanted to dig into this is also within BV-BRC, we have alpha fold structures. And so you can look at where the SNPs are occurring on the structure. These little colored asterisks are my annotations of what's happening on the structure. So this is what it looks like, and that's what we're gonna look at. And you can even dig in more deeply to see those. So that's what we're gonna do in this series of videos, and I really hope you enjoy it. And if you get a chance to read the paper and watch the YouTube video for that particular study, it is so awesome. Okay, let's get started.